Hi, my name is Chris Mancuso. I'm a postdoc at Michigan State University. Today, I'll be talking about jointly embedding networks from multiple species to improve gene classification. A couple years ago, we benchmarked a method that uses supervised learning. It uses the network connections. It represents the network connections somehow as different ways as features inside of supervised machine learning model. And we extensively benched this mark against the state-of-the-art kind of go-to method of label propagation for network-based gene classification. And in general, we showed over many different kinds of tasks, function, disease, trait prediction with a bunch of different networks, that supervised learning generally always outperforms label propagation. So if you go to that link there, that is the benchmarking paper. So the new thing that we're trying to do is if you can take information from multiple species to improve gene classification, this gene classification method, which we call gene plexus. So for instance, so you can take a human and mouse network, and then you can connect nodes together in the network based on orthology. And you can use a couple different networks like BioGrid and IMP, any networks that have kind of information for multiple species. And so we took a collection of model species. And so one way you can do it is you can just make the connections and then just concatenate, put all of the nodes in one thing. So then you have a pretty large matrix. But if you do this for many networks, this gets really big, really fast. So you can also embed these, each node in a low dimensional vector. And we use Node2Vec or SVD to do this. So what we did is we tried to really extensively see how building these cross-species models networks how we can optimize it. So there's using the adjacency matrix, where we just concatenate the edges directly. Then we can take that adjacency matrix and spread the information on the edges with random walk with restart. We can use Node2Vec to do random walk based lower dimensional representation, and then also just SVD. So we can connect the nodes together based on just, if it's a connection, we give it a weight of 1. We can use the degree of the nodes that are connecting it. We connect it directly node to node, or we can make a new node for an orthologous group. Tried a couple different embedding sizes. With random start with rewalk, you can change the restart parameter. You can use the transpose of the matrix as features. SVD, we can standard scale it or not scale it. So kind of like SVD and kind of PCA-ish. And then Node2Vec has a lot more parameters. You can do depth first walking, breadth first walking, number of walks, walk length, context window size. But really only Node2Vec scales well to having many species together. So this is kind of like an example of an extensive benchmarking that we did, is that this is all the different methods, and then there's all the parameters, and this is kind of picking like the best method out of the parameter set. And we can generally see that combining multiple species, combining the human network and the mouse network together makes Go annotations in human and mouse much better. Another thing is that we tried to kind of do the same thing, but for a number of different other model species. So what we have here is the orange again is all species networks. And then the blues are human and the model species. And then the green is just the model species networks. And it's predicting Go annotations in the model species. And we can see that adding the human and mouse almost always is better. And Node2Vec is a pretty great way of capturing this information. And then another thing we did is just purely train models on human genes and then predict genes in the other species. And so for here, you have to have the human and mouse model, because this is kind of the big thing that, like you can put these genes in the same space now. And so what we can do, and what you can see here is that doing this with Node2Vec, this is for the BioGrid network. So doing this with Node2Vec is much better than just concaten using the adjacency matrix. And the bottom row here is like kind of counting. So if you predict on a Go term, then you see in the set of Go terms in the other species that you predict, which one, if there's a matching one, is that matching one kind of have a higher evaluation metric than all the other Go terms and the model species. So it does. So that's just a little bit about the poster. I will be in front of my poster, or you can email me, Twitter me, or whatever. And then if you scan this will take links to all the kind of gene plexus papers that we have. Thank you very much. I hope to see everyone at my poster. Thank you.